Good afternoon. My name is Adam Cohen. Along with Michael Lynn, I am one of the co-hosts this afternoon. Welcome to the session on emerging high-throughput microscopy methods for imaging brain activity. Recent advances have dramatically improved the resolution in time, space, and throughput of brain imaging tools. Today, we will hear about these exciting developments from some of the leaders in the field.
And my name is Michael Lynn. I'd like to reiterate the welcome. Thank you all for joining us. So we have a very exciting program today, so I won't keep you guys away from it too much longer. But just two reminders before we get started. First is if you will see at the bottom of your screen that you can submit questions to the speakers. You can submit those at any time. We are going to go through them at the very end of the session. That is about an hour from now, but you can submit them at any time. And if you could indicate if there's a particular speaker who your question is directed to, if you could just type their name at the beginning of your question so we know who to direct the question to. And we'll be organizing and fielding those questions at the very end. And so we have a very exciting lineup with Valentina Emiliani, Meng Cui, Sarah Abramson, Jerome Mertz, Kasper Podorsky, Nazi, and Alapasha Vaziri. And we will get started. Thank you very much.
First of all, I would like to thank Adam and Michael for having organized this virtual symposium, and I wish that everybody in the audience stays healthy and safe during these challenging times. So the interest of my lab is to develop and use advanced optical methods for the manipulation of neuronal circuits using wavefront shaping and optogenetics. As you probably know, conventional experiments using optogenetics today are done using visible light wide field illumination, and the cell type specificity is reached because of the precision of genetic targeting. So with this approach, it has been possible to achieve a very precise mapping of brain functioning with cell type specificity. However, the use of wide field illumination can only synchronously activate or silence entire population of cells, while looking at the activity of circuits under physiological condition, we know that in most of the case this is characterized by the fact that even genetically identical cells can have a very divergent activity pattern. So today, if you want to start manipulating those kind of circuits and eventually even mimic their activity with the ultimate goal of being able to link specific spatial temporal activity patterns to behavior, we need somehow to replace the whole region optogenetics to what we called a few years ago circuits optogenetics, meaning with this a combination of techniques that enable to manipulate single or multiple targets independently in space and time. So in this talk, I will show you quickly a quick overview of which have been the most important technical challenges that has to be faced to reach the condition to perform circuits optogenetic. So first of all, we need, of course, to get cellular resolution, and for that we need to replace single photon wide field illumination with focus to photon excitation, so to be able to reach a single target embedded in a three-dimensional scattering volume. And to increase the efficiency of two-photon optogenetics, in my lab we have proposed to combine two approaches with two-photon excitation. The first one is called computer-generated holography. So here the key element is a spatial lane modulator that enable to modify the propagation of the light so that at the sample plane you can get an illumination shape that eventually precisely reproduces the shape of the original target. And to also get precision with respect to the axial propagation, we propose to combine computer-generated holography with the technique of temporal focusing, where now the key element is a dispersive grading, which enables to to excite the sample with a laser beam having a short pulse selectively at the focal plane, and so therefore with optical sectioning independently on the lateral spot shape. So we were able to show that the use of the combination of those two approaches enable a very efficient current integration when used to excite cells expressing an optogenetic channel. And for example, when this is combined with different opsin in vivo, we shown that parallel illumination enabled to reduce the illumination time to generate a single action potential down to 5 milliseconds. And because we can keep the illumination time so short, we could also reach sub-millisecond temporal jittering. Means that we can very precisely control the spike in time. We then also shown that the use of parallel illumination is compatible with the use of Opsin with a fast photocycles, and this has the advantage that they can be used to trigger action potential train a very high spiking rate, up to 80-100 Hz. So this is as far as concerns the temporal resolution. Then the next important step was to control the spatial resolution, and for that there have been in the past year many labs that has developed different variants of what we call soma-restricted opsin, which have the characteristic of expressing the opsin selectively only into the soma. And therefore, when these are combined with targeted illumination, they enable to reach a true single cell resolution. So an example of that is shown here, where I'm showing the recording taken from the layer 2-3 of the visual cortex in vivo, where we first choose those 3 targets for photostimulation. These are the corresponding calcium responses, and then we selectively only choose one of the 3 targets, and as you can see here, again from the calcium responses, this shown that we were able to select only the activity of this specific target. So the next important step was then to be able to perform this kind of manipulation eventually by controlling the activity of many targets and distributed in a 3-dimensional volume. So this required to modify the optical system, and what we propose is to do so by using 2 independent spatial layer modulators. A first one creating generating an illumination shape at the grating for temporal focusing, and the second one which is used to multiplex this shape in 3D at multiple locations. So this optical system, as is shown here, then admitted multiple variants. For example, this first part can be replaced by an expanded Gaussian beam, and then we call the technique called 3D shot, or this can also be replaced with alternative shaping approach, for example, the generalized phase contrast method, or ad hoc variants of amplitude phase modulation techniques. So for a lack of time, I won't have time to go through all of these approaches. You can see them described in this reference. But here, let me just show you an example where multi-target is achieved by using twice an holographic system. So this is shown here. So the first spatial layer modulator create the holographic disk, which is then temporally focused, and the second one, as is shown here, multiplex this temporally focused disk in 3D, in this case 50 different locations, 50 different axial planes. And because each of these disks is temporally focused, the axial resolution was kept below the 12 micrometer in the entire field of view, and which is the right size we need to get through a single cell resolution. So we then show that we have shown that the wavefront shaping that enable to perform a 3-dimensional light generation is also compatible with the propagation through a green lens, and so this is shown here, and where you can see that actually it is possible to create multi-temporally focused shapes through a green lens. And so if this kind of optical system is combined with a system for 2-photon imaging, is it then possible to perform at the same time manipulation and calcium imaging in depth? So the very last point I would like to discuss now is what about the temperature rise induced when we increase the number of targets that we want to illuminate. To answer to this question, we developed a few years ago a model and to evaluate the local temperature rise induced under the typical condition to perform two-photon optogenetics using a single spot or multiple spot. And as you can see here, these are the results of the simulations, and interesting what we learn from this is that if the illumination time is kept before 3-5 milliseconds and the average distance among the multiple targets is kept larger than the corresponding thermal diffusion length, that for this illumination time will be of the order of 50-60 micrometer, then the total temperature rise at each location can be kept below the reported damage, thermal damage threshold. And this model then was also validated experimentally using erbium with erbium co-doped glass particle, and we demonstrated there was a good agreement between what the model was expecting and the experiments, and this now is was also recently done directly in vivo. So it is time to conclude, and the general conclusion I would just like to say is that today a combination of different approaches, which include illumination approaches, but also Opsin and reporter engineering, modeling, and laser source development, put us on the way to manipulate in a very precise way neuronal circuits, and therefore to perform these what I defined circuits optogenetics. I would like with this also to thank now the people of my lab, of the Wavefront Engineering Microscopy Group, the many collaborators and the financing source, especially the NIH through the BRAIN initiative. And I would like, of course, to thank you for your attention, and with this also to introduce my the next speaker, Dr. Meng Kui, who is going to talk just after me. Thank you for your attention.
The research is compact. It's really about high-throughput deep brain imaging. Even with the advance of multiphoton microscopy, the imaging depth in mammalian brain is typically about 1 millimeter. So the large portion of the mouse brain is not accessible, not to mention larger mammals. In the past, miniature probe has been utilized. For example, green lens and a microprism and their combination. A challenge of using such probe is the very small imaging area. Green lens is it has a very simple structure. If you use interferometry to quantify the spatial aberration, you will find there's a strong position-dependent astigmatism. As a result, the two-photon field of view is very small. This is really like imaging through a tunnel. You can only get information from the tip, only in the middle regions. All the other area is not accessible. So 2 years ago, I have a chance to visit Atlanta Aquarium. They have a nice underwater viewing gallery. Walking along this gallery, you can look around this gallery to see all the fishes. So you have actually 2 degrees of freedom. One is walking along this gallery, the other is looking around. So this inspired me to come up with a very simple solution. I always said that a 5-year-old kid can come up with the solution. Instead of inserting probe into brain tissue, you actually insert a capillary. The capillary is only 15 micron thin. So inside the capillary, you fill index matching liquid, for example, water, to match the index to the brain tissue, and then you insert your imaging probe that actually can spin around and also moving up and down. In this way, all the tissues around this capillary become accessible. To test this idea, we started with two-photon microscopy. So the idea is really using similar system as conventional method, but just adding SOM. This is to compensating the spherical aberration of the green lens and also controlling the focal depth outside the capillary. We have a rotary stage that can spin and translate this imaging probe. Using such control, we can access all the tissues around this capillary. So in this way, we can acquire massive amount of information. Just using this simple probe, we actually can achieve That means we only have this green lens and the prism. We can achieve decent spatial resolution, about 1.2, 1.3 transverse resolution, actually 14.8. The field of view can be quite large. If you image actually 400 micron outside the capillary, you spin around, you have 6 millimeter field of view from this 1 millimeter probe. So you can acquire lots amount of information. If you utilize better compound lens, that means you have some more components underneath the green lens, you can achieve better resolution. For example, 1 micron. X-ray resolution is about 10 micron. So this is excellent for imaging soma and also dendrite. The key of this experiment is in the surgery and the mechanical alignment and control side. For surgery, we tested several methods and we arrived at one solution that's very robust. This method is first to insert an open-end capillary into the brain tissue. As the capillary is only 15 microns thin, this is actually thinner than most razor edge. So you can cut this into brain tissue. We use a vacuum pump to remove tissue inside, and then we put in a closed-end capillary at 10 micron per second, controlled by a motorized actuator. And in the end, we have a cap that's designed to match the head bar to prevent dust coming into this head bar. After 4 weeks, we can start function imaging. That is assisted by 3 cameras, 1, 2 for transverse alignment. This is 1 for vertical. With the 3 machine vision camera, we can do this alignment within 2 to 3 minutes. The key advantage is really we have a massive amount of tissue accessible. For example, we can measure a large amount of tissue volume all the way from cortex to hippocampus. If you have time, you can really visit a huge amount of neural population. A key consideration for longitudinal study is to revisit the same neurons over time. In other words, we need to find the same neurons. For example, we did an experiment in which we load the animal at different time points, and then we command the system to visit the same location, same orientation. We can easily find the same neuron, for example, here and here. There are some tens of a micron offset. I think eventually, if we utilize machine vision to reset position 0 after the insertion, we can actually reduce this error down to 10 micron level. For calcium imaging, the current mode we are doing is so-called park-and image. Essentially, the probe is stopped at one location. We record some calcium data and move to another location. And of course, there's even faster approach possible. We are also working on the so-called spinning and imaging. That way, we can acquire large population at the same time. The signal-noise ratio is actually excellent, not only from the soma, but also from the dendrites. One thing we are thinking is how do we broadly disseminate this method so many labs can start using it. One solution we come up with is very simple. We call this thing O-ring method. Instead of changing anything to your existing 2-photon system, you just have this capillary. You have an O-ring that can actually put onto this probe. By adjusting the O-ring's position, we can determine how far we want to go inside this capillary. And by controlling the orientation, we can determine which region we want to image. Therefore, without changing imaging system, we can achieve some of the function of a compact system. Our goal is to have a neuroscientist start using this method. And if you find this is really helpful, we can help you guys motorize the system, make the system very user-friendly. In the end, the key message is that compact offer near 3 orders of magnitude tissue access volume. A huge amount of tissue can be accessible for high-resolution imaging. Essentially, what we did is that we're using this capillary to isolate brain tissue from the imaging probe. And therefore, we convert a deep brain imaging problem into a high-resolution fluorescence endoscope. And there has been a lot of great invention over the past 2 decades that we can take advantage of. In summary, compact is good for both 1-photon and 2-photon imaging. It's a minor change to surgical procedure. In the past, we insert a green lens, now insert capillary. We can gain huge tissue access volume. There is great flexibility. In the past, if the neurons of interest are not there, you need to have a new animal, new surgery. Now you can really freely explore large population to find a neuron of interest. The other thing is that we can even change the probe inside. For example, we can switch between high-resolution probe to resolve synaptic structures and another probe for really large volume recording. So we can take one probe out and put another one in freely because we have the capillary inside. It's through machine vision camera and also the robotic surgery, we can have the system very robust and reproducible. There are lots of things to be done to make the speed even faster. Miniaturization, the other one way is to make it really small to access small brain region. The other is really make it large to access the whole brain of monkeys. We want to have very high resolution up to N8.7 for resolving synaptic structures, all the way from head fixed to free moving animal and also from mice to monkeys. The goal is really to have whole brain, mammalian brain, neurophotonic access so that we can perform optical mesoscale deep brain recording and control at any regions inside the mammalian brain. I'd like to send many of our collaborators and also funding from Purdue startup and also equipment from HHMI and also funding from NIH and the Brain Institute project. If you have questions, feel free to contact me. The next speaker is Sarah Abrahamson from UC Santa Cruz.
My name is Sarah Abrahamson. I'm an assistant professor at the University of California, Santa Cruz. I've been here for a little over 2 years, and I've set up an optical systems design laboratory. Today I'll talk to you about our latest invention, which is the 25-camera multifocus microscope, which is intended for use in neurobiology to look at the very fast spiking dynamics by recording large 3D volumes at over 100 frames per second. You see on this slide a prototype of our first M25 instrument. This prototype was built at the Marine Biological Laboratory at Woods Hole in the lab of my collaborator Jennifer Morgan, who studies lamprey. And what you see also on this first slide is a 25 focal plane image of the lamprey spinal cord with fluorescently labeled neurons. There's this conflict between 3D resolution and 3D acquisition speed in microscopy that we are sidestepping using diffractive Fourier optics methods in order to obtain ultra-fast images of 3D volumes with either the optimal resolution that the microscope is capable of or even super resolution. But I won't talk about super resolution today because it's not quite as fast. So that's not what we're doing with this M25 system. Thinking about imaging, the basic image that the photographic camera or the microscope or our individual eye forms is a 2D image of a 3D reality. So here we see from the F64 group a photograph from the High Sierra with very, very deep depth of focus. So we can actually see the entire 3D scene in this image, but this is not a very useful mode in all biology because we cannot tell where anything is in this image. So instead, in microscopy, we open up our aperture and we get an image with very high resolution, both laterally and axially, meaning we have a very short depth of focus. And we can really see the image plane only. On cherry blossom in Central Park here, this is not as powerful as in microscopy. In microscopy, we have a semi-transparent sample and we have fluorescent label that we look at, which means that we can really look through them and gather the entire 3D volume. This example is the nuclear pore complex of a mouse fibroblast nucleus. So this focal stack is recorded in a multifocus microscope. So that is the raw data on the camera chip. Normally in fluorescence microscopy and white field microscopy, you would scan through the entire volume of the cell and record 1, 2, 3, 4, 5, 6, 7, 8, 9, or however many focal planes you needed to cover that volume. Here we record all the information at the same time to get truly simultaneous 3D imaging. And then we can go very, very fast and we have no ambiguity in time between the different focal planes that we record. So the way we do this is through diffractive optics, which is my favorite microscopy or favorite optics. We all know in reflection in the mirror, light reflects off the mirror. And in refraction through a lens, the lens passes through the lens and is refracted. In diffractive optics, we can actually multiplex the beam, and we can also change the phase of that beam so that we can use a diffractive optic device, which is a single piece of glass that's about this size and has a pattern of little steps in that glass etched into it. And by controlling that pattern very, very well, when the light passes through it, the light ripples just like a wave. So this is sort of tapping into the wave nature of light. And by designing that grating absolutely perfectly for our application, we can obtain that instant focal series. The way we do that is, first imagine that we put a periodic grating in the Fourier plane of the microscope. The Fourier plane is the back focal plane of the objective, and the microscope consists of the objective lens and a tube lens that together form an image on the camera. So now we pop that little grating in the back focal plane of the objective, and that multiplexes the image. We also wanted to focus shift these images. So what we do then is that we adjust the pattern on that little optic so that it has tinier periods of the grating pattern on top and bigger spacing on bottom, which affects the diffractive angle. So looking at that in the microscope, we have the objective, we have the back focal plane, we place the optic here, and our multiplex beams, if we have a periodic grating now, have the same diffraction angle. Now that we introduce a chirp to that grating, we have a larger diffractive angle on top and a smaller on bottom. And looking at what that does to the wavefront of the light, we see that it introduces a defocus. So the +1 order, which we call the diffractive order that goes up, it's actually defocused. So it will not focus on the camera here, it will focus somewhere else. And the -1 order, which is the order that heads downward, is converging. So it focuses before the nominal focal plane. And if we adjust that pattern of this optic perfectly, we can actually make a perfect focus image of every plane in the sample. So here is an example of 3 planes. We normally would make this so that we have a structure that scatters light also left and right, so that we get that 9 image planes. So here's that again. And when we do this, we use the Abbe-Sine condition to calculate how we completely remove the out of focus phase error of each one of those focal planes. And then we adjust the Z-step or the focus step so that it fits our sample. Sometimes you want to sample a little finer and sometimes a little coarser in Z. Here's the free open source code in MATLAB that you can download if you want to design your own diffractive optics. This is a pattern that generates 9 planes. You can also generate multi-layer patterns. Those have higher photon count efficiency and can be much more efficient in your imaging system, but they're also much harder to make. So there's a give and take there on how much time you want to spend in the clean room. Thankfully, my students love spending time in the clean room, and it's really fun, and they can make their own gratings. This is a picture from the nanotech facility in UC Santa Barbara that we use a lot. Once we've made these optics, we build easy to use, user-friendly imaging systems and commission them. This is 2-arm, 2-color multi-focus microscope for imaging transcription in yeast. And this is our student Robert Shalansky who's been working on that. But what we're really interested here is neurobiology. So this is the key 2 set of neurons in the C. elegans animal. This is data from when I did my postdoc in the laboratory of Corey Bargman at Rockefeller University. And we see these sensory input neurons, which are input from smell, reacting to the smell of almonds. So even this powerful, this method with only 9 planes was powerful enough that we had to completely change the way data analysis was being done in the lab, because students could no longer manually annotate this neural signaling. So we worked with Liam Paninsky's group to use intelligent algorithms to automate this data analysis. What we really wanted, though, was to be able to image all the neurons simultaneously. So I started working on this larger volume 25-plane microscope, and I put all of those images on the same camera chip. By then, CMOS cameras was pretty new technology, but it was still not quite enough to get the high resolution in sampling that we wanted. So today, we have even better technology at our hands. We have these small, fast, sensitive CMOS cameras, and I was able to redesign the multifocus microscope layout to this, which only uses this one diffractive grating, and then another diffractive grating, which sits in front of the lens of each individual camera, and produces a very corrected image of 25 focal planes at the same time. So here's example data from that. This is GCamp muscle in the nematode C. elegans and neurons that have been injection labeled with calcium dye in the lamprey spinal cord. And that here is our first movie of the C. elegans. This is nematodes from Gal Haspel, and it's taken in Jennifer Morgan's lab at the Woods Hole by my PhD student Eduardo Hirata. And we just can't wait now until this lockdown stops so that we can start employing this prototype and also the prototype that we built in my lab at UC Santa Cruz to start looking into those fast dynamics at faster than 100 frames per second in these large 3D volumes of living samples. Here's my lab, my students. Thank you very much. It was a pleasure to be here virtually.
So I'd like to talk about a technique we've been developing called reverberation multiphoton microscopy to address three key challenges. We'd like to do high-speed imaging inside large volumes of thick samples such as brain tissue. Well, as we all know, the standard way to image deep inside brain tissue is to use multiphoton microscopy, which is basically a scanning microscope with a pulsed laser. But standard multiphoton microscopy only produces single plane imaging. This can be extended to two planes using a technique of spatiotemporal multiplexing that was first demonstrated by the Jeff Squire group over a decade ago. And the idea here is to split an illumination beam into two paths, one path being longer than the other, such that an incoming pulse here is split into two pulselets separated by a few nanoseconds. And if a lens is inserted in one of the paths, then both pulselets will focus to different depths inside the sample. And in this way, a single detector can look at both depths essentially simultaneously, and to discriminate these depths, all you need is a fast enough detector with nanosecond time resolution. But that's fairly standard these days. So this can be extended to more than two depths, but very quickly it gets very complicated. We found that a simple way to do this is to insert what we call a reverberation loop inside the illumination path, such that now an incoming pulse gets split into basically an infinite series of outgoing pulselets. And if in addition we add a lens inside this loop, then every time the pulse circulates through this loop, it revisits the same lens, meaning that each one of these pulselets focuses to a different depth inside the sample. But as you can see, since we're using a 50-50 beam splitter here, each pulselet has a different amount of energy, which decays according to this power law, where each pulselet has half the power of the preceding pulselet. And the first question one might ask is, does this pose a problem that we're using different powers to irradiate each plane? And to answer that question, you have to remember that we're looking inside a scattering sample. And so it actually works up because, as we all know, as you increase imaging depth inside the sample, you actually need more and more power with multiphoton microscopy. And in fact, things can be arranged such that if you choose this interplane separation just right, then the fluorescence coming from each plane can be made nominally the same. And the optimal interplane separation to do this is given by this very simple formula where delta z, the optimal separation, is the scattering length of the sample times the log of 2, which for brain tissue corresponds to about delta z of 100 microns. So here's some initial results. This is simultaneous 6-plane imaging, vascular imaging inside the mouse brain in vivo, going all the way down to about 600 microns. And what you just saw was a z-stack where all we did was scan in z from one plane to the next, and all the other planes sort of followed so that we could look at the entire volume here. And each frame sees all 6 planes simultaneously. Color here corresponds to depth. For example, this is a diving vessel right here. And we have a fairly large field of view, almost a millimeter, and we're going video rate. Here's calcium imaging, 3-plane calcium imaging. This is GCamp looking inside the mouse olfactory bulb. And this is evoked odor response, where when we puff on an odor, some of these cells light up, some of them actually get dimmer. And in this manner, you can look at even the same cell, the dynamics of the same cell across multiple layers inside the brain. So a question we often get is, what about photodamage? It seems like we may be dumping a lot of power in the sample. So to demonstrate that it's really not that bad, we cranked up the power by a factor of 3 compared to what we normally use, and we continuously imaged for about an hour and a half, and we showed that the response of the cells was basically unchanged. So photodamage doesn't seem to be a huge issue here. So why is that? Well, part of the reason is that there's an inherent advantage to distributing your planes in the axial direction. For example, for a given amount of power, if you achieve single plane imaging to a maximum depth penetration with standard multiphoton microscopy, then with the same amount of power, you can achieve multiplane imaging all the way up to the sample surface using reverberation multiphoton microscopy. And the only hit you took was a decrease in the maximum depth penetration by this distance delta z. And if you want to achieve the same depth penetration as standard multiphoton microscopy, all you have to do is double the power, and that's independent of the number of planes that you're looking at all the way to the sample surface. So here are some of the advantages of reverberation multiphoton microscopy. But it does have one small disadvantage, and that's that the planes are a bit further apart than we would like. They're about 100 microns now. So how did we address this problem? Well, here's a schematic of the actual reverberation loop. And again, the way it works is that a pulse going in gets split into a sequence of pulselets coming out that are distributed in time. But they're also distributed in space, because this loop, which is basically a 4F relay, is adjusted to be a little bit too long, such that each pulselet coming out is coming out at a slightly different axial position, which means that there will be a separation between where they focus of delta z. And we can adjust that separation by adjusting this loop length such that this separation in the sample is about 100 microns. But again, 100 microns is a little bit too long. We found a way to half that distance by introducing a second loop, which we call the interleave loop. Now here the pulse only goes around once because we're using a polarizing beam splitter. So what happens here is an incoming pulse gets split into only 2 pulselets that are slightly delayed in time and space. And each one of these pulselets then creates their own sequence of reverberation pulses that are interleaved, both in space and time. And in this way, we achieve half the interplane separation. And importantly, there's no power loss in the system, and everything remains collinear. So in summary, with interleaved reverberation multiphoton, we can achieve a 50 micron interplane separation. But we thought even that was a little bit too wide. So what we did was we added a piezo stepper on our objective so that we dithered the objective up and down by only 25 microns. This halves the net imaging rate, but now we achieve a 25 micron interplane separation. And our future plans are to actually go gapless by introducing quasi-Bessel beam illumination. And as a benefit, this adds robustness to aberrations. So that's our future plan. So here are some results. This is 10-plane imaging in the zebrafish. This was taken at 15 hertz using a Menlo system laser that we were able to demo. This has lower repetition rate than normal, which is good for reverb. This was even lower repetition rate. This is a coherent fidelity laser with a lot of power, albeit in the red. And here we're looking at cardiac organoids. And finally, this is just to show you what our software looks like. This is what we see on the computer screen in real time as we're scanning around. This is 18-plane imaging at 50 hertz. This is just beads. So with that, I would like to thank everyone you see here, in particular Devin Beaulieu, who was basically the workhorse behind this project, as well as all the funding sources. Thank you very much. And finally, I would also like to introduce the next speaker, Kaspar Podgorski from HHMI Janelia Farm.
Hello, everyone. I really appreciate the invitation, and I'm very happy to be here. My name is Kaspar, and I'm a fellow at HHMI's Janelia Research Campus. Today, I want to highlight a distinction that deserves more emphasis in our community. That's the distinction between studying neural coding, meaning the information contained in neural firing patterns, like the receptive fields, and neural computation, meaning how that information is transformed from inputs into outputs as it flows through a neural circuit. This is essentially a distinction between what and how. I don't just want to know what a neuron responds to, I want to know how it's able to respond that way, the quote-unquote logic of how it operates. We're already very good at measuring the what. We can easily measure spikes in response to stimuli or correlate them to behavior. But it's harder to get at the how. To study the logic of neural computation, we need to know which neurons communicate to which other neurons and the patterns of signals they send. New methods are emerging that are going to make it easier to study computation, such as indicators that report subthreshold communication between neurons. But the tools we currently have for measuring neural activity are still much better at studying these things on the left column than in the right. And to get better at studying neural computation, we're still going to need to put a lot of work into making tools. My lab's approach to studying computation is not to try to record for more neurons or record the same number of neurons faster. Expanding the bandwidth of somatic recordings is very important, but I just don't think these types of recordings are especially helpful for studying computation. Every neuron here receives thousands of inputs, and most of them come from other brain areas. Hidden input dominates the activity of every neuron we record, creating latent variables that confound our models. Recording more neurons does very little to solve that problem, so this approach, regardless of scale, makes it really hard to study things like learning rules or other basic concepts from the theory of neuroscience. So instead, my lab is working on tools to record comprehensively from single neurons. If we could record many inputs from across a neuron's dendrites and its output, we could start to ask questions about how that neuron transforms information. And unlike somatic imaging, where any given pair of neurons we record is probably not connected, every synapse that we record onto a cell represents one of that cell's connections. I think that single neurons and their interfaces are the right way to carve nature at its joints to study neural computation. And I think that goal deserves as much attention as recording from more neurons faster. So what do we need in order to make comprehensive in vivo recordings of single neurons a reality? First off, we need microscopes capable of high-resolution recording from large numbers of synapses at hundreds of hertz to see patterns of activity on the timescale of neuronal signal integration. Our approach to that problem has been a new type of two-photon microscope that uses line excitation to collect multiple 1D projections of the 2D sample plane. We scan lines across the sample at 4 different angles, so this is a form of compressive tomography, and it's closely related to CT imaging. The 4 lines here scan the sample in less than a millisecond, so this kind of imaging is extremely fast. By using a structural image of the sample as a prior, we can record activity within that structure from these high-speed compressed measurements. This approach overcomes physical limits on how fast sequential point scanning microscopy can go. On the left is a 2-photon recording of fluorescent particles flowing through a 100-plane volume, imaged at 1.5 billion voxels per second. On the right is glutamate imaging of neuronal dendrites, containing hundreds of synapses recorded at over 1 kilohertz. This type of imaging fundamentally requires deconvolution to go from the 1D measurements to the 2D image space. So some of our work has been on the development of efficient optimization algorithms that perform deconvolution subject to the kind of spatial and temporal priors that are available for brain activity imaging. These are things like knowing the structure of the sample in advance, as well as the dynamics of the indicator and its maximum response amplitude. We've built on some of the concepts from that first microscope to build a second one that's more flexible, is better at 3D imaging, and can also perform richly patterned optogenetics. This microscope is designed specifically for interrogating entire dendritic arbors of mouse cortical neurons in vivo. The new microscope works by scanning a line of excitation across a DMD and having the DMD select which parts of the line get relayed to the sample. The great thing about using a DMD like this is that we can change the pattern on it extremely quickly, and shape light differently for every sweep of the excitation line. This gives us random access to large numbers of target sites without the access time issues of conventional random access scanners. When we image activity, we're often trying to measure the brightness of regions of interest, like the dendritic spines on this image on the left. On the right here is a random pattern of 500 such integration ROEs. This is how our new microscope records those 500 ROEs. It sweeps the vertical line of excitation across the DMD 20,000 times a second. Each laser pulse illuminates a column of the DMD. If I pause this, you can see that only one ROE is open in any given column at a time, meaning that we only measure one ROE at a time, so you don't need computational unmixing to see your data. It also means that sparsity is not required for this type of imaging. The microscope has two of these scan fields that can be steered to different locations in the sample, or overlaid to image one plane twice as fast. It records those 500 ROEs from the last slide at a mean rate of 3.6 kilohertz each. And because the measurements are independent across ROEs, we can record the activity of those ROEs as fast, we can recover the activity as fast as we record them. Another thing that comprehensive single neuron imaging will need is better indicators. So I've been lucky to have Eric Schreiter and Lauren Luger's labs and the Genie project just upstairs from me. So we've helped them test some of their new indicators for GABA, acetylcholine, and membrane potential. My lab has also made a yellow variant of GCAM, which is efficiently excited by 1030 nanometer fiber lasers that are useful for these types of high-speed imaging systems. Most recently, my lab has taken on producing a new generation of glutamate indicators in a project spearheaded by Abhi Agarwal. Calcium indicators can report synaptic activity, but they're much more often swamped by backpropagating action potentials, which is one big motivation to try to use neurotransmitter indicators to record synaptic activity. But neurotransmitter indicators have their own limitations, and there's still a lot of work to do to understand the signals they convey. One issue with GluSniffer is that many variants sense glutamate spillover. These are tuning maps where brightness encodes response amplitude and saturation encodes selectivity. Glutamate signals are on the left, and spine calcium responses are on the right. This GluSniffer variant shows relatively non-specific signals in a lot of membrane structures, indicating spillover. Using expansion microscopy, we found that the existing membrane targeting vector of iGluSniffer is excluded from postsynaptic densities in cortex. And in general, there's a variety of properties we'd like to tune for all neurotransmitter sensors, like membrane trafficking, subcellular localization, affinity, and kinetics. And of course, their brightness, sensitivity, and photostability, which can vary between 1 photon and 2 photon excitation. We've been working to improve all of these properties of GluSniffer. So on the left here are spectra in the presence and absence of glutamate in vitro for superfolder GluSniffer, the previous variant, and a new variant in yellow. On the right is the 2 photon delta F over F. Abhi's evolved this sensor using high throughput screening under 2 photon illumination, and it has especially large responses in 2p compared to 1p. This is 1 photon imaging in culture. The new sensors are darker in the dark state, brighter in the bright state, and more efficient at getting to the membrane. So far, we've only done a little bit of testing in vivo, and we plan to continue optimizing variants for specific experiment types for a few more months. In summary, I think we should look to single neurons to answer many important questions about how the brain computes. My lab is working on methods to make such studies possible, so we're developing microscopes, we're working on improving indicators, and other things that I've only touched on briefly, like theory and computational tools for making inferences from these kinds of measurements. Thanks a lot for listening. And I want to thank our collaborators and the wonderful community that makes it so fun to work in this field. And if you want to hear more about what we're working on, please reach out to me. Our next speaker will be Naji from the University of California, Berkeley.
Thank you, Casper, for the introduction. Today, I will be telling you about our recent result using kilohertz 2-photon fluorescence microscopy for in vivo voltage imaging. Raster scanning 2-photon fluorescence microscopy is the most popular in vivo brain imaging method. Because the fluorescence is only generated around the focal volume of an excitation beam, in order to form an image, typically this beam is scanned in 2 dimensions in the raster pattern, and the generated fluorescent signal detected by a non-imaging detector such as the photo-multiplier tube to form a 2-dimensional image of the sample. This method has very high spatial resolution, allowing structures as small as individual synapses to be detected. And it can also be applied at depths hundreds of microns into the optically opaque brain. However, because of the focus is typically scanned across the sample using mechanical devices, its temporal resolution is not fast enough in order to track the very fast membrane potential change of neurons. In order to observe those events using optical method, we now require a kilohertz raster scanning 2-photon fluorescence microscopy method. And we built such a microscope using an ultra-fast line scanning method called free space angular chirp enhancement delay, or FACET, in collaboration with Kevin Sha's lab at University of Hong Kong, who initially proposed this method. We built a 2-photon microscope that is capable of 1,000 frames per second imaging. It is based on a conventional 2-photon microscope that is equipped with a set of galvo scanners and that can scan the excitation focus across the sample. The FACET unit itself is a passive optical device made of 3 components, 1 cylindrical lens and a pair of high reflectivity mirrors that are almost parallel to each other except for a very small angle alpha. If we look at now the red beamlets, after it was launched into the pair of mirrors, if the mirrors are strictly parallel, it will just keep reflecting upon them until it exits from the other side of the mirror pair. But because of the very small angle alpha, the incidence angle keeps decreasing until it will hit one of the mirror perpendicularly. At this point, it will retroreflect and then can be redirected into the microscope and form a focus. The neat thing about this module is that now if you look at the dark gray beamlet, and what you can see is that it enters the mirror pair at a smaller incidence angle. As a result, it takes a fewer number of reflections before it will become perpendicular to one of the mirror and then retroreflected and enter the microscope at a different angle, which means that now this light pulse will be focused to a different spatial location. And these pulses, in addition to being spatially separated, they are also temporally delayed so that we can distinguish the fluorescence that is being generated by them. So optically, what is happening is that this pair of mirrors basically formed an array of virtual images of this focal point, which was then imaged into the mouse brain. So in this way, it is very similar to take an image of an object in between 2 mirrors. However, with this very simple device, what it does is that it converts a single pulse, which will form a single focus, into an array of sub-pulses that are temporally separated and spatially separated as well. And in other words, the FACET unit can scan the focus in 1 dimension at the laser repetition rate, which in our case is 1 megahertz. In order to form a 2-dimensional image, we just need to scan using the conventional galvo along the orthogonal direction. And even using a conventional galvo, it's very easy to achieve 1,000 frames per second. Using this microscope and the new genetically encoded calcium sensor, genetically encoded voltage sensor developed by the Micheal Lin lab at Stanford, we finally were able to detect both sub-threshold as well as the super-threshold or action potential response of neurons in vivo, as shown in this example. Because we are using 2-photon excitation and non-imaging detection, we will also be able to image very deep within the scattering brain. Here we are looking at 4 example neurons at 4 different depths from the same brain. And then you can see from the superficial neuron all the way down to a neuron beyond 300 micron deep, we are able to see both the action potential as well as the sub-threshold events. Looking at the response to individual action potentials, we characterized the rise time, fall time, as well as the delta F over F value for individual action potentials. And these values are consistent with those characterized in vitro for those sensors. And another advantage of 2-photon excitation non-imaging detection is that we do not worry about crosstalk. all the signal contamination between neighboring neurons. As you can see here, the 4 neurons that were active within this same field of view has a distinct activity pattern without any sign of crosstalk. We also explored the sensory evoked activity from awake mouse. Here we are looking at a neuron within the primary visual cortex of the mouse, which is selectively active towards drifting grating of particular orientations. So when we present the mouse with a non-preferred grating, you can see that there's very little activity in terms of the membrane voltage change, either subthreshold or superthreshold. But when the cell is presented with its preferred stimulus, we see a very active action potential firing as well as the subthreshold voltage change. And we can extract both the action potential spiking response as well as the subthreshold response and use that to calculate the tuning curve. And as you can see, the action potential based tuning curve, which is black, is a lot sharper than the subthreshold response based tuning curve due to the thresholding effect of action potential firing. This is consistent to the result obtained with whole cell recording electrophysiological method. And we also look at the onset time of the sub as well as superthreshold voltage response after the start of visual stimulus presentation. So here we have 2 neurons at 345 micron below the surface of the brain. And on top, these 2 traces, it's a subthreshold response. As you can see, about, in this case, 50 milliseconds after the onset of the drifting grating stimulus, we start to observe the subthreshold response. And a very similar value was obtained for its action potential firing as well. So here is the data we obtained over 20 cells, which again consistently showing about between 50 to 60 milliseconds of onset time for the subthreshold response. And this method is very robust, and we were able to get many visually evoked neurons from awake mouse. And here we see a very similar action potential onset time of 60 milliseconds, consistent with previous measurement using whole cell as well as cell-attached recording. So in summary, using a passive optical module based on the concept of facet, we converted a conventional 2-photon microscope into a kilohertz frame rate raster scanning 2-photon system, which allow us to measure both the sub as well as super threshold voltage response in vivo. So this work is led by Daniel Wu, a postdoc in my lab, and previously a postdoc in Kevin Sha's lab at the University of Hong Kong. Another collaborator on this project is Micheal Lin's lab at Stanford University. Thank you very much for your attention. Next speaker is Anipasha Varizi from Rockefeller University.
I will be talking about a few techniques that we have recently developed in the lab that are aimed at recording the large-scale neuroactivity in a volumetric fashion in the scattering rodent brain. As you heard from previous talks and you're all aware, the standard tool for recording neuroactivity in the scattering brain is 2-photon scanning microscopy. However, I want to highlight some of the limitations that are associated with this technique if it's applied to a volumetric acquisition technique. Imagine that we have a volume that is about 500 cubed in size, and this would be the size of our diffraction limited acquisition. And we want to sample this volume at about 5 Hertz. So simple math shows us that this translates to about 500 million voxels, which sampled at 5 Hertz means that we have to have an acquisition rate of about 2.5 gigahertz. And since we have to excite each voxel at least with 1 laser pulse, we need to have a laser that runs at that rate. Now, this has 2 problems. First of all, a laser at that repetition rate is not commonly available, but even if it would be, it means that the dwell time at each of these voxels is about an order of magnitude shorter than the fluorescence lifetime. So we will not be able to collect any fluorescence light out of these voxels. So we need to think about it in a conceptually different way. And one question we ask is, can we relax the spatial resolution without losing information? Now, if we look at the size of typical mouse cortical neurons, which are about 10 to 15 micron in size, we find that perhaps the resolution of the diffraction limited excitation spot is perhaps unnecessarily high, at least when it comes to imaging neuronal cell bodies. So we can easily underfill the microscope objective and thereby create a larger excitation spot. But the problem with that is that if we do that, we lose all axial localization or sectioning capability of our tool. So we will not be able to distinguish neurons that are on top or below each other. So from this, it becomes clear that we need to be able to create a comparable lateral and axial resolution, and which requires us to be able to decouple these parameters of the beam, and that needs us to do light sculpting. Now, one simple technique for doing light sculpting that we and others have used in the past is the technique of temporal focusing. So the basic idea here is that we can utilize the bandwidth that is associated with these femtosecond pulses to control the axial localization of excitation. And the way that can be done is by sending these pulses to a dispersive element such as a grating, from where then the different spectral components diverge in space. And then the process can be reversed by using a telescope that consists of a lens and our microscope objective, which is configured such to be bringing all these spectral components together only in one axial location within the sample. So while we can achieve the axial localization of excitation by using dispersion, the lateral localization can still be achieved by the original size on the grating and the demagnification ratio of the telescope. So doing that, we created basically an excitation spot that was isotropic, about 5 micron lateral and 5 micron axial, and then we can scan that quickly using standard ways for raster scanning in X, Y, and Z. Now, going back to the example that I started, the volume now translates to about 1 million pixels sampled at 5 Hertz, and using 1 pulse per pixel, which turns out to be the most effective way how you can create fluorescence for the same amount of average power, means that we need to have a laser that at least runs at 5 megahertz, which is more conventionally available. Also, our pixel dwell time is now only 200 nanosecond, which is sufficient to collect all the fluorescence we need. So we implemented this scanned temporal focusing approach and a couple of additional ways how we could further speed up the voxel acquisition rate. One of them was this idea of remote focusing, basically by rapidly changing the divergence of the beam into the backfocal aperture by creating a conjugate plane to the sample in where we can place a light-weighted mirror on a fast scanning voice coil. We can create a scheme that allows us to rapidly scan along the Z dimension. We also implemented the idea of temporal multiplexing. The basic idea here is basically we can use time tagging of pulses, as you heard in one of the previous talks, by delaying the pulses by different amounts and identifying them uniquely through the time of arrival in the sample. And then these pulses can be then directed to different axial locations or lateral locations within the sample, which can be all scanned at the same time and the fluorescence collected on the PMT. So using this strategy, we first implemented the axial multiplexing and looked at the mouse PPC. So here you can see now a volume that is about 700 by 700 by 600 microns, containing on the order of 6,000 neurons, and we can sample here the whole volume at about 17 Hertz. We can also reconfigure the volume such that these subvolumes are next to each other. Thereby, we can create a much larger lateral extent. In this case, we looked at the primary auditory cortex of the mouse. So you can see here now a volume that is about 1 millimeter by 1 millimeter by 0.6 millimeter in size, containing about 12,000 neurons, which can be all sampled at about 5 Hertz. While this shows the activity, if you look at the typical traces of these neurons in this heat map representation, the sheer density and number of these makes it very difficult to see the activities. But if you zoom in here into about 300 of these, we find that they exhibit quite well characterized individual time series of neurons as we know them from typical calcium traces. And in this case, we could see the responses of individual neurons to different frequencies between 6 kilohertz and 80 kilohertz, with single neurons responding to individual frequencies with some cross responses to neighboring auditory frequencies. So this showed us that we can use this approach to do effective large-scale and volumetric recording using 2P, but eventually, as we go deeper into the sample, the signal-to-background ratio starts to drop and reaches unity around 800 to 1000 micrometer. So at that level, the only thing that helps is to switch to a higher nonlinearity by introducing, for instance, 3-photon excitation. So to look at the deeper layers of the brain, we introduced also 3-photon. So in this case, we implemented a second path that using a second laser light that was all synchronized with our original 2-photon pulse train, but had an independent path in which we had individual galvos, dispersion compensation, where we also implemented similar optimizations in terms of point spread function engineering and single pulse per pixel excitation, as I highlighted for the 2-photon case. And doing that, we can now look and go through all the layers of the cortex, all the way to the corpus callosum, and reach into the C1 neurons of the hippocampus. Here you can see a single plane recording at about 1.16 millimeter depth with the individual traces of the neurons here. And here a single plane recording at 1.22 millimeter image through intact cortex at about 78 hertz, which you can see exhibits still very nice signal to background ratio and dynamics of the individual neurons. One could say maybe the speed is unnecessarily high given the response time of GCamp, so we can use the excess time to image instead in a volumetric fashion, which is shown here from a depth of 750 microns to about 1000 microns. Here we are looking at about 400 neurons that we are sampling now at about 4 hertz. Finally, we can also do a hybrid realization in which we reserve the more power intensive 3-photon excitation for the deeper layers and use 2-photon volumetric excitation for the upper layers. So here you can see now simultaneously recorded activity of all the cortical layers doing 2-photon sculpted light excitation with 2-photon, while doing simultaneously volumetric recording at the deeper layers using 3-photon excitation. So here we can collect from a volume that is about 700 by 700 by 1100 microns, containing about 3,500 neurons. And you can imagine that this tool can be quite useful in telling you now information about correlations within different cortical layers with respect to each other while the animal is engaged in various behaviors, but also correlations between cortical and subcortical layers of the brain. And I'm happy to announce that as of this year, this technology has been licensed by 3i Imaging, and they will be coming out with a commercial solution that would be available. Thank you.
I don't know if everyone can hear me. We're going to get started on a Q&A. Yes, okay, so I think we're online now for the Q&A session. So thank you very much for everybody who has submitted questions. We have a long list of questions. So what we'll do is I will repeat the questions that were asked by text, and then the designated target of that question can answer. I'll do this for first few speakers, and then we'll switch duties. Adam and I will switch duties. So we have the question for Valentina from Jahida Basa. Hi, Jahida. And the question is, how deep can you perform photostimulation with your method? And now, Valentina, if you are there, you can unmute yourself. Valentina may have stepped out for a second. We can get back to Valentina. The next set of questions are for Mansui. So a lot of people are interested in your technique. We had lots of technical questions about it. I'll just go through these real quick. One person was curious about the resolution provided by the technique. Another wanted to know what the upper frame rate would be. The third person wanted to know about the cost. and whether it would work in freely moving animals. And another person wanted to know about compatibility with other modalities such as spinning disk or structured illumination. And I see that you were considering answers to the questions by text, but if you could tell the entire audience what you think in response to these questions, that would be great.
Okay, so I'll first maybe starting from the resolution one. The resolution is limiting by the imaging probe. So we tested two different imaging probes. One probe is very simple. It's just bonding a small prism to the bottom of the green lens. That one has about 1.2, 1.3 transverse resolution, 14.8 axial resolution. And the other probe we tested is a compound lens. That is a green lens. There's some miniature lenses and also followed by a small prism. For that one, we can reduce the axial resolution to 10 micron, transverse resolution to 1 micron. Such resolution is okay for soma and dendrite, but if you want to really resolve synaptic structure, you need an even higher NA. We are actually working on a higher NA probe that's NA.7, but it's still under development. Let's see. Another question related is about cost. So we're also thinking about how can we really broadly disseminate this method. So one thing we propose, and I'll also show in the slide, is so-called O-ring method. With that method, you don't really need to change your imaging system. Essentially, you follow the procedure of inserting capillary, and then you just have an O-ring on your probe. Then you drop this probe inside the capillary. That allow you to control how far you go into the capillary, and also you need to control the orientation. Of course, this is quite a manual involved, but the cost is really low. You can less than $1,000. But if you really want to motorize the system, that involve purchasing a rotary stage and also a few small cameras, I think the cost will be a few $1,000s. And right now, the imaging we do is so-called park image. So we park the probe at one location, take some measurement and do that. It's not really fast enough. So another module we are working on is called spinning while imaging. That will make it faster, but it will also make the system a little bit more expensive. But I think it's on the order of $10,000. That's the cost. Let's see.
But you were saying that in terms of the speed and compatibility, it's basically whatever you want to put into the system, right? You were saying that you can use a resonant galvo, so it's just a standard.
Yes, I think it's always limited by how much signal you can get. I think with the miniature probe, one thing challenging is it's not as big as the conventional optical lens, so we won't be able to collect a fluorescent signal over a very large entendure. So your signal compared to conventional, imaging non-invasively, your signal will be less. So this actually set the fundamental limit. But if you really just want to push for higher frame rate, of course you can switch to resonant galvo, try to get it faster. That's the fundamental limit. The other thing, I think, with the spinning and the imaging, you can also cover a larger volume simultaneously.
Yes. Then to follow up on that fundamental limit, you were saying it's less sensitive because you're collecting from a smaller area. But if you had a really big capillary, that's the size of that's a full width of a field of view, wouldn't that?
Yes, I think if you really, for example, for some of the probe we listed, the small one is from 350 all the way to 1 millimeter and 2 millimeter. If you can really push to 2 millimeter, then I think, for example, for monkey brain and larger animals, then the collection will be rather efficient. Really, you have a rather large entendure even compared to the conventional commercial available optical lenses, then it's really going to be efficient. But for the small, if you really want to miniaturize the device, push it to small diameter, that's an important factor limiting how much signal can get. Yeah.
Great, thank you, Meng. So we're going to move on to questions for Sarah. So we have some questions for you, Sarah, from the audience. Well, one is from Adam himself, another is from Jerome, and a third is from me. So we're curious how your technique compares to light field microscopy. And then I was curious about the sensitivity of these CMOS cameras, like these cheap CMOS cameras, as we bought some and we thought that they were going to be really good based on their specs, but they didn't seem that great.
Yeah, I saw these questions, and I actually put text answers on them. You gotta be quiet now, Lucas. The first one is light field, which is super cool, right? However, it does compromise resolution, and it's inherent. I sat down with Mark Lavoie and Michael Brackison a lot and tried to compute it, and there's no way to do it that we've thought of that doesn't compromise resolution. The main problem with light field, though, has been the computation that you need to reconstruct the data, because it's not deterministic when the sample is transparent. So really, it is good to get a full resolution, just regular 2D image that you don't need to guess what it is. When it comes to the light efficiency, which someone also asked, we lose less than 50% with our optics, which is as good as you will ever get, because we have optimized everything completely with fabrication and design. So you up to a 50% loss you will take through these diffractive optics that we use. After that, it's the same as a 2D image, but you have to remember, when you take a focal stack, you take that image multiple times, and so do we, which means that if you compare to 2D imaging, you multiply by the number of focal planes you have. When it comes to the cameras, they are noisier. This is the problem. They already have 70% quantum efficiency, which is not that much less than the 95 that the newest CMOS chips have, which are really, really great also, which we have two of that we're testing out this summer, side by side with the EMCCDs. But subtracting the dark image helps a lot, actually. So just cover your aperture, take 16 images, take the average of those, and subtract that from every frame in your data. And after that, I think computational denoising is super exciting. One of my PhD students is working exclusively almost on that. And Joshua Batson at the BioHub has been super helpful with this. And computational denoising is going to be, I think, super, super important. The photon shot noise is the ultimate limit, right? But as long as we have any electronic noise, we should first work to remove that.
Right. And Jerome wanted to know.
Different ones of those cameras are differently good. So the IMX 73 and 249 are super awesome from Sony.
Oh, great. I'm writing that down. Okay. So, and then Jerome had a question what the delta Z was. I guess the Z resolution. Oh, Sarah. Jerome had a question about the Z resolution.
Oh, yeah. It's the same as wide field. Yeah, same as wide field. Oh, how deep we can go, the range. So we are not sure, because what's going to limit us is when we're going to start to see clipping inside the objective. We've gone to 50 micron depth. That's the deepest we've done. We're going to try 100 next. And since it's all design of the interior of the objective is proprietary, we can't calculate it. We're going to have to try it until we start clipping. Because what happens is that you have those planes, but they got to enter the objective. And at some point, we'll have vignetting, and we don't know when. So we're going to try 100 microns next. And after that, if that works, we're going to try 200, and we'll see what happens.
But Jerome, was your question about the maximum depth or the resolution in the Z?
Resolution is the same as wide field.
No, it was the maximum depth separation, the maximum separation between planes.
Yeah. So we've done 2 micron in the 50 plane, and we've made optics for 4 micron, and we can't wait to get back in lab so we can try them out.
Great. Thank you, Sarah. So let's see if we have questions for Casper. Oh, wait, we're going to go back to Valentina. I don't know if Valentina is back. She may not be back. So I will ask Casper's questions for Casper, and then we'll turn it over to Adam. So so Tom Kirkland from Promega. Hi, Tom, has a question about which neurotransmitters are most important to image for functional imaging of the brain. And I had a question for Casper about how the DMDs scan 500 regions of interest at 3 kilohertz. Casper, you are on.
Hi. Sure, I'll start with Tom's question. Hi, Tom. I don't know that there's any authority that makes the list that you propose. I'm definitely not the most qualified person to be making the list. If there's something that you're specifically interested in, I really suggest getting in touch with some of these folks that make these things, like Lauren Luger or Lin Chan or Yulong Li. Obviously, glutamate and GABA, we have indicators for. Those are sort of the most common or the most prevalent excitatory and inhibitory neurotransmitters in vertebrates. In invertebrates, acetylcholine is very important. But if you're working on physiology, perhaps the most interesting things are some of these lesser known indicators. And so there's a lot of really exciting work on neuropeptides and neuromodulators, some of these transmitters that might have previously been considered sort of secondary, like glycine or D-serine. I think the neuromodulators are really especially exciting because they control so much computational state in these circuits. Everyone has their own sort of wish list. Definitely worth having that conversation with folks that are developing new indicators. Michael, your question is about how you how you can target many different independent sites. So the DMD access time is something like 50 microseconds, but that's to refresh the entire image on the DMD. And the measurements are actually made much faster. They're made at the repetition rate of the laser. So you have, every laser pulse is a measurement. And so we make measurements with a line that's scanning across a DMD. So every measurement is one of these columns that then gets sort of sub-selected. We throw away everything except a column of an ROI in this sort of integration rowy mode that I talked the most about. And so we're making measurements actually at 41 megahertz in this case. And there's really very little dead time in this case, because if you have lots of ROIs open in the field of view, most of your time is spent measuring an ROI. So if you think of sort of the time efficiency of this type of measurement, which is, the important criterion if you want to talk about how many different ROIs you can measure, it's very, very good and much better than sort of the access times that you could get with AOD type scanners, because the access time becomes the laser repetition rate in some cases.
Right, I see. So basically, you're using the DMD as a mask with little holes in it, and then you're putting a laser line across it quickly, like this, right?
Yeah, and Adam's lab has done sort of a holographic analog to this recently with an SLM, where they addressed rows or columns of an SLM and then had a 1D hologram. These are actually conceptually there's sort of similarities in this approach where you combine the sort of programmability of an SLM or a DMD with the sort of the fast access time of scanning a laser to sort of a line to a spatially distinct point. And so that's what gives you the fast access time.
That's great. That's very elegant. And then there's one more question, which is from Jaida. And that is, do you have an automated segmentation algorithm for dendritic ROIs? And have you compared the performance of GCAM7B with iGlue sniffer?
Yes. Okay. So there's two questions. Automated segmentation is extremely important. So in SLAP, which is that tomographic mode, tomographic microscope that I first briefly introduced, you don't have to do segmentation in advance. You can do it after the fact. And so it can be automatic or it can be manual. You can really do whatever you like. We developed automatic methods there that are just based on Elastic is a random forest classifier that you can train easily. More recently, because with this new mode where you do integration rowy imaging, it's really important to rapidly do instant segmentation of in vivo images. There we've used a sort of UNet style design followed by some post-processing to sort of agglomerate and identify synapses. And that's something that we're working on, and we really want to make that as turnkey and as widely usable as possible. I think a very important thing to work on. Sorry, Michael, do you remember the second question?
It was what if you compare GCAM7 and iGlue sniffer? And actually, before you answer that, we have a question from Doug Kim, who's one of the conference organizers. Hello, Doug. He wants to know if anyone should buy 1030 nanometer lasers and what color sensors and Opsin's everyone should make now. I guess the question is whether we should all try to convert to 1030.
This is a super important discussion topic, I feel. I don't know.
Yeah, that's right. So I don't think we have time to cover all those things, but if you can answer those questions briefly, that would be great.
Sure. So the imaging that I introduced, because we do this very parallel excitation where, at least before these sub-selections that we do, we have to put a lot of power into a lot of points at once. I really like using fiber lasers to do this. And we actually saw fiber lasers in a couple other places. Ali Pasha showed a 1030 laser, as did Jerome, or 1040. And so these lasers are great because they're very stable and they generate a lot of pulse power that's hard to do at sort of the classic GCamp, GFP excitation wavelengths. And so we recently made a variant of GCamp that is yellow. We've changed it to a YFP chromophore, which took a little bit of work, and that allows us to excite it very well with those fiber lasers. It's a wavelength that is really great for exciting RFPs as well. So there's great arguments for using maybe a combination of yellow and red fluorescent proteins with these sort of inexpensive and powerful fiber lasers. There's also, great semiconductor lasers at those wavelengths as well. So we don't get great SNR out of GCamp because GCamp excites about 14 times more poorly at 1030 than it does at its peak. We have done imaging with GCamp. GCamp is a great sensor, so you can sort of overcome that. You can take that hit. But it was really great for us to move to yellow indicators. And glue sniffer is very bright, especially under 2 photon. Gives great signals, and we're only working on improving some of the sort of challenges for using it for synaptic signaling.
Great. I'm going to turn it over to Adam.
Thanks.
Great. So there are a couple of questions for Naji. One of them from my former grad student, Lin Fan, is how long can you use the facet approach with the ASAP voltage indicator? I guess that's a question related to photo bleaching rates. And then the second question is, could you resolve any functional dynamics down to subcellular structures using the facet technique with ASAP?
Those are two great questions. So for the ASAP sensor that in our paper, which is ASAP 3B, we were able to image in a cell for a cumulative 80 seconds. And it wasn't completely bleached. We stopped the experiment because we didn't need to image it more. But these 80 seconds are not continuous. So we image for 1 second, and then we wait for 2.5 seconds. What we found is that the brightness of the fluorophore actually go back almost completely. Now, before the shutdown, we were actually testing new sensors from the main lab, which in the preliminary testing, we actually didn't see any photo bleaching within the limited amount of exposure. It's possible that the basal fluorescence is quite a bit dimmer in that sensor. So I'm very hopeful that this wouldn't be a limitation. And it's already not a limitation, even with 3D, for many experiments that people may be interested in. So the second question is about, sorry, my cat was trying to attack me. So can you remind me what's the second question?
It was about resolving subcellular voltage dynamics.
Yes, right. That's really the ultimate goal is if we can see all the membrane depolarization on individual synapses, that would be great. With the current sensor, it's not. We don't have that sensitivity to see it yet, but that's ultimately the goal.
Great, thank you. Another question for you, Na, is: are the individual pulselets that come from the facet technique equivalent in power and other optical properties, or is there some drift as you go across the array?
Right, so the pulses differs by how many times they get reflected on those mirrors. And the mirror has very high reflectivity. It's higher than 99.9%. So even from the first to the last pulses, their energy difference would be less than 8%, even if the reflectivity is only 99.9. And the mirror also has very low group velocity dispersion, which means that those pulses also wouldn't differ in terms of how long they are. The main thing that distinguishes the pulses, especially from the very first one to the very last one, is a beam size. Because light diffracts, the more you propagate it, the size will get bigger. So in the figure 1 of our paper, we actually characterize the point square function of individual pulses. So basically, the most delayed pulse has a higher effective numeric aperture because it fills the back aperture of objective more. But that's really the main difference among the pulses.
Great, Thank you. Maybe we'll end with a question which is to all of the speakers. So we'll let you speak in whatever order you like. Is about data management. And but the blessing and the curse of high throughput microscopy is that you have large quantities of data coming in at high speed. Is there any hope for common analysis or handling software, or does everybody have to write their own software? And what are people doing about that challenge? So, since you answered the last question, maybe you want to tackle that one first?
Yeah. So with FACET, we do have very high data rate, and it would be great if we can all have the same kind of data connection pipeline. But practically, just like in all the different methods described here, we are taking data in very different ways. Like either raw data coming in, for Jerome's case, it's going to be at different depths, and in my case, it will be at different lateral position. So in the initial stage of data analysis, I do not see how a universal pipeline would apply to everybody. But then, of course, once we use FPGA or other methods, we can get the reduce the data size. Then many of those data are basically going to be exactly the same, just metrics representing two-dimensional, three-dimensional information. And with that, we basically use whatever that is already existing. Data analysis pipeline. Many of them are open sources, and we found them to be very useful for us.
So Ali Pasha, I'm curious what your lab is doing with, since you spoke specifically about these huge data rates that these experiments can generate.
Sure, yeah. I mean, I think as a general comment, I think what would be probably more useful in my view would be to develop something that would be more like a common platform, not necessarily a common data processing pipeline. It has to have features to be adaptable to the different differences in these imaging modalities. So we specifically have been very successfully using the Cayman that was developed by Liam Paninsky's lab, because it has certain parameters that we could modify and match and optimize for our particular imaging modality. And otherwise, I think having the infrastructure in place for shipping data, over networks at high speed, because just copy paste becomes not such a simple task. So having all of this infrastructure is also another important thing that we implemented a long time ago, both in terms of processing and also transfer rates.
Great, thank you. So maybe I'll end coming back to Valentina, because I think she's back now on the audio. The question for you, Valentina, was how deep can you perform the photo stimulation? Her audio still seems to be not working. Okay. In that case, I think we should thank all of the speakers for their presentations and for answering the questions, and to thank all of you in the internet, in the ether, for coming to this session, and wish you all the best with your research. Thank you.
Yes, thank you, everyone. Thanks to all the speakers and the audience.